Hi, good evening. So it's 8 p.m. on the 16th of May 2021, and this is a look back at the news from Zimmer and Peacock for this week. So every week we like to do a sort of retrospective look at the week and just sort of highlight some of the news that we've put out from Zimmer and Peacock. And we always have a really busy kind of week, so I'll try and go through it in the next few minutes or the next few, yeah. So just some of the news that we put out there this week. We have a Generation 2 hydrogen peroxide sensor. So at Zimmer and Peacock, you know that we have lots of biosensors: glucose, lactate, oxygen, sodium, potassium, enzyme-based, ionophore-based, and we also do a lot of immunosensors as well, where we have antibodies binding to antigens. One of the famous sort of sensors that we also have is this COVID-19 sensor. Anyway, so we have a lot of sensors. The hydrogen peroxide sensor is one of the ones we've had for the longest, and we've described it here as a Generation 2 hydrogen peroxide sensor. Now the reason we call it Generation 2 hydrogen peroxide sensor is because if you look at glucose sensors, there was a Generation 1 glucose sensor where the enzyme reacted directly with the oxygen and gave you hydrogen peroxide. And so the oxygen there is really known as the sort of native cofactor. Now with the Generation 2 glucose sensor, people started changing away from oxygen and using redox mediators like ferrocene and ferricyanide. So when we describe our Generation 2 hydrogen peroxide sensor, what we're meaning by that is it's not it's a hydrogen peroxide sensor, it's enzyme based, but it's not using oxygen as the mediator. We're actually using a redox mediator. And this allows us to be able to detect hydrogen peroxide at lower voltages, which means that we can be more specific because we have less background interference. So a little bit of news from Zimmer and Peacock this week. We put some data out about our Generation 2 hydrogen peroxide sensors. I think we very, very rarely talk about these hydrogen peroxide sensors, but we've been making them for quite a long time now. And because we put some more data out there, we got some more data this week, I thought we'd just put it up onto the blog. So at Zimmer and Peacock, we make glucose and lactate and oxygen and alcohol, uric acid. Oh, and by the way, we also do hydrogen peroxide. And hydrogen peroxide is actually really relevant at the moment because a lot of people are using, they call fogging machines. So in hospitals, in places where they want to make sure that there's no COVID-19 on the surfaces or in the atmosphere, they're using fogging machines. These fogging machines are actually producing hydrogen peroxide in the vapor phase. And so there's actually an increasing interest in hydrogen peroxide sensors. So we make them for often medical applications, more specifically measuring hydrogen peroxide in fluids that are associated with the humans. But we are getting a lot of inquiries these days because people are making these fogging machines which produce hydrogen peroxide. They sort of flood a room full of it and try and kill all the viruses. So that's why hydrogen peroxide sensors are becoming really popular at the moment. So I'm just going to move on slightly now and just talk about some other news from Zimmer and Peacock this week. We did put a little bit of a note out there about biosensors and their applications. There are review articles on biosensors, but I think some of the more popular ones I see are kind of around from sort of 2016 and today's 2021. So biosensors for us, we play them into the food industry. So if you're not aware, then Zimmer Peacock does a suite of sensors for the food quality industry. The most famous one we do is a chili sensor for measuring the hotness of chilies. So, when doing a review on biosensors, food industry is really important. Another one that's really important for us is biosensors are being used in agricultural. I think this is one of the most significant biosensors that we're working on, is measuring the nitrate in soil. The reason I think this is significant is because nitrate is added to the soil for good reason, so that we get good plant growth. But of course, unfortunately, it runs off the soil, runs into the groundwater, or runs into rivers, and can cause essentially pollution, because it over fertilizes the water courses. You can have algae blooms and overgrowth of vegetation. So we did a little review article, and we did touch upon biosensors for food, biosensors for agriculture, and then we also, of course, talked about biosensors for medical applications, which is what we do an awful lot of, including that hydrogen peroxide sensor that I just mentioned. And I think with the biosensors, you have enzyme-based biosensors. Glucose oxidase is the kind of grandfather of all sort of biosensing, let's say. In a glucose sensor, you have bioinfinity, that's sort of immunosensors where you have an antibody binding with an antigen. DNA is also a form of sort of bioinfinity, where you have a single strand of DNA and it attaches to another strand of DNA. We do a lot of ionophore ion selective electrodes at Zimmer and Peacock. One that we've not done so much of, but I'm actually super intrigued by, is actually microbial biosensors, using a culture of microbes as the sensing element. We haven't done a lot of that, but I'm personally quite interested in that. So we did this kind of review, and we just talked about the applications and then the types of biosensors, and we put some links around to kind of give people examples of biosensors. It's probably worth knowing that, one of the missions at Zimmer and Peacock is to, we want to be a successful business. Of course, every business does want to be, but we also want to make sure that we're doing the right thing globally, and that's why we kind of try and put out a lot of, free information as well, because we believe that one of the futures of mankind and humanity is going to be biosensors, and biosensors are going to play an important role in the future of us all. So we're quite happy to put out, the kind of information, and we put a lot of effort into it. Now, some other news that Zimmer and Peacock put out there this week was regarding AI. So I was pleased, I'm delighted that many of you attend the ZP Developers Zone every Thursday at 8 AM London time. We do a whole video, or a whole webinar rather, about the Developers Zone. And this week we talked about Julie. Julie is a cloud database system that we have. It's free, so in that cloud database people can go into the lab, they can gather their data, they can upload it to Julie, and they can start doing analysis of their data. They can actually share their data as well. And ZP is constantly adding new features to Julie. Now, what Julie at ZP allows us to do, it allows us to complete part of our mission, which is to take tests that are otherwise in the lab, HPLC, and move them to be low cost point of care or point of need tests. So in the webinar that we did this Thursday, we talked about how we're trying to reduce all these large clinical analyzers down to something that's quite small and hopefully low cost. And then we talked about one of the strategies we have at Zimmer and Peacock is we want to develop assays, as economically and as quickly as possible. So, for example, if you're a food scientist and you have a way of testing a particular analyte, let's say it was a sort of porcine analyte, and you said, right, I want to make a sensor so that I can sort of ensure that this product doesn't have gelatin in it or something, that's something derived from from pigs, for example. Then you often have maybe an HPLC method or a reference method, something that's quite it's something that's something quite bulky in HPLC system, but that's a reference method. Now, what we like to do is we like to take that reference method and we like to take a sample and we like to run that sample on the reference method, so you have a real value, something you really believe in. Now, what we're also trying to do at the same time then at ZP is we're trying to develop a biosensor as well, and we develop biosensors that fit into these little bits of hardware and have a sensor associated with them as well. So we're trying to develop a biosensor, but at the same time, it's really great if a reference method exists as well. Because what we're able to do is we have a database called Julie. It exists in the cloud. So we're able to actually send data to Julie along with the reference value, and we actually can compare our biosensor data versus the real, let's say, most accurate reference value, and we can tell we can make our algorithm better. We know the real value is the reference value. We get a value from the sensor. If those two things are not as correlated as we like, then we can actually use Julie and the AI algorithms in Julie to actually improve the algorithm. And it's possible because we have the sensor, the reference method, and we're very good at app development, and we have the Julie database. I just got asked a really good question actually as we were live streaming by Ali, who says, can a biosensor tell the difference between live organisms and dead organisms? And it was a good question, and actually I think the quick answer might be this. Just brainstorming. I appreciate the question as well. Ali has a PhD in this kind of subjects as well, so we're on the same wavelength. And the answer is, of course, microbes often are taking up oxygen. So I think you can tell whether a microbe is alive or dead using a technique like, I believe, impedance spectroscopy. So I have seen people use impedance spectroscopy to interrogate microorganisms and see if their cell walls are intact. And when the cell walls are not intact, I believe it really changes the impedance spectroscopy. And I say that's one way of doing it. I think the other way of doing it is actually looking at the oxygen consumption. Because essentially, if I have an electrode and it's acting as an oxygen sensor, and you can do that by applying minus 650 millivolts, for example, to an oxygen sensor. When the microbes are there and they're living, essentially they're probably consuming most of the oxygen, and so the electrode is in this kind of, it's in an anaerobic state because all the oxygen's gone. But of course, as soon as the microbes are on their way out, the oxygen flux possibly will start increasing, and you'll get a jump in oxygen peak. So the reason I suddenly changed gears then is because we do this news as a live stream, and a question came through, and that's a good one. Can electrochemistry to tell between live and dead microorganisms? And I have definitely read, there's quite a body of work on impedance spectroscopy for this. And then as I was thinking about it as well, I was also thinking you could probably do it through the oxygen signal. I actually think it's good to do both. I always like, if you can look at a problem in two ways, maybe impedance and maybe chronoamperometry, looking at the oxygen, then you can say, if these two things are telling me the same answer, then we would use our data science to sort of say, two things tell me the same thing, it's therefore correct. So we had our webinar and we talked about how AI is assisting us in biosensor development, and I think maybe I'll just finish at that, not finish it, but on this point, AI is really important. So anyone who's doing biosensor development these days, if you're not learning Python, please start learning Python. There's some pretty cool stuff going on in the world of AI, and at ZP we're definitely part of it. We put a little note out there this week as well about a sort of holistic approach to biosensors. At ZP we've got a large team now, and it is growing, and we're looking forward to growing it even more. And people will come to us and say, can you make me and it's very specific, I want a screen printed gold electrode, and that's all they want from us. And that's completely fine. We understand, that people don't want sometimes all of the things we can do, but, a biosensor is not just about the screen printed electrode, or it's not just about the sputtered gold electrode. It's about the electrode, it's about the microfluidics, it's about the quality system, i.e. the sort of reproducibility, it's about the manufacturing, it's about the regulatory requirements, it's about the microfluidics, bring the sample to the sensor. So we just put a bit of a page up there because we do, people do come to us very specific and they say, I want just this off you. But these are all elements that come together. And it's actually, it's not a good idea just to go to a vendor and say, I just want you to do this one small part of the problem, because these parts are all interrelated. And so you sort of, with maybe a company like ZP, it's actually let us get involved in many of the things, and because together, all these things come together, and you actually end up with a really quite good product. But at ZP, we get involved in helping people with market research, proof of principle studies, contract development, ISO 13485 consulting. I'll say it again, ISO 13485. This is just really important for anyone who's doing biosensor development. We do the contract biosensor development. We do the clean room assembly. We do clinical support. We literally do, not literally, we absolutely do go into hospitals and train the nurses or get the patient samples. And then there's really important part is, microfluidics. A big part of getting a reproducible signal is bringing a sample to a sensor in a reproducible fashion. And, many people really don't think about microfluidics too much, but at ZP we definitely think about the microfluidics. So we do have a holistic approach to biosensor development. You can't just think about one part, but you have to think about the whole problem. We had a really good discussion this week in the developer zone, and one of the subjects that we covered was actually temperature control. Sorry, not temperature control, but temperature effect on enzymes. And we ended up getting quite deep into the maths on it. In chemistry, there's something called the Arrhenius equation. It's like many things in life, it's an approximation. But we did discuss that when you look at this temperature effect of a biosensor, it would indicate that the temperature effect was probably due to the enzyme kinetics. And we did, a fairly sort of logical, explanation as to that. Doesn't make us right. It just means that the theory hasn't been disproved yet, but at the same time, we haven't proved it either. But at Zimmer and Peacock, we are a we're a pragmatic company. We want to make the best biosensors we possibly can, but we also know the limitations of what we're doing. If you want to have really in-depth analysis of a problem and absolute certainty, then my academic colleagues are definitely the places to go. But at Zimmer and Peacock, we want to make a sensor, we want to make it in a reproducible way, we want it to work, but sometimes there's sort of, the vagaries of whether my signal is diffusion controlled or enzyme limited. These are interesting, but they're not our primary purpose. But we do like, when people ask the questions, we will dig in and and try and get in deep on those things. And just as an interest as well, I did get an inquiry this evening for people asking, should we have an undergraduate's part of the developer zone? So the developer zone is this kind of free website. We do these webinars, and maybe we should actually start thinking about having content specifically for undergraduates as well, because at ZP we're a big believer in biosensors being really good for humanity. And we need to encourage, the next generation of developers. So if people think it's a good idea to have an undergraduate zone in our developer zone, let me know and we can work upon it. Now, rather interestingly, we do get inquiries about gold sputtered electrodes. So I just want to say to people, that there's lots of forms of gold that you can use in biosensor development. Gold is often used in immunosensors, where you have an antibody and you want to bind it to your surface, and you might do it through a thiol molecule. But at ZP we've got at least four types of gold. We've got gold sputtered on glass, gold sputtered on polymer, PCB, which is printed circuit board gold. We've got screen printed gold. When it comes to screen printed gold, we got two variants there. We've got high temperature gold, which is often 800 degrees Celsius, and low temperature gold, which has been cured at 150 degrees Celsius. So that's about 5 versions of gold that we've got there. The reason I put that up is because we'll have somebody say, oh, I want this gold, very specific. But again, it's my point earlier on that, when you're doing biosensor development, you have to, people go to somebody like Zimmer, people say, I want this, it's very specific what I want. But actually, I think with somebody like ZP, tell us what you want to achieve, because the goal that you may have chosen or think, might not be as manufacturable, it might not be at the cost that you need. So sort of tell us a little bit about what you want to achieve, and then we can help you, advise you, is that gold functionally good for you, and will it meet the price points that you need. So I know I said ISO 13485 quite a bit this week, but we are an ISO 13485 company. We follow quality systems. If you're used to working in a university situation, it can feel a little bit different. But I would say it's not stifling. I mean, at ZP, we do lots of interesting work. I think we have a lot of fun. But we are, an ISO 13485 company. So when we consult on this. I mean, it's interesting because when you look at the ISO 13485 products that we have these document templates. And I don't know how many document templates we have on our website, but this is probably a fraction of all the kind of documentation that it requires to make a biosensor. We've got documents here for technical reports, project development, process requirements, IQ, OQ, PQ, CAPA, NDAs, corrective actions. Yeah, corrective actions. I mentioned CAPAs already. Non-conformances. There's a lot of documentation that goes into biosensor development, and I think it's kind of, I do try to encourage people to reach out to us at ZP, because we do generally try to offer people placements. We sponsor postdocs through the KTN, the Knowledge Transfer Network, the KTP, the Knowledge Transfer Partnership in the UK. We have a PhD available still in Norway for doing biosensors for fish. So I do encourage people, follow us, get in contact with us. If you can do a placement with us, it can lead to full-time employment. All that said, and one of the things you'll surprise when you get to us is actually, is the structure, is the amount, is the documentation that we have to implement, because many of these products are going to go to market, and they might even be involved in human health. One of the documents that we have in our quality system is a risk assessment. What's the risk of a sensor giving an inaccurate answer? I mean, one of the biggest sensors, biosensors on the market, is obviously this glucose sensor, glucose sensors for diabetics. If you accidentally report somebody, for example, that their glucose is 5 millimolar, in fact it's 1 millimolar, that can unfortunately lead to a coma and death. So it's really important, when you're doing biosensor development and you're really thinking about going to market, that you have to be working in a quality system. And one of the first things the quality system is going to ask you is, have you done a risk assessment? What's the risk of something going wrong? And I just gave you an example, severe example there. And that's why when I was talking earlier on about the applications of biosensors, I sometimes quite like food quality, because the risk of miscalling the hotness of a chili is not as, let's say, risky as miscalling the glucose for a diabetic. So some applications are more forgiving on error than other applications. So finally, I said that at Zimmer Peacock we have to have this kind of holistic view on biosensor development, and it's actually, what stops most people getting biosensors to the market in the end is actually, is essentially money. Is there really a business plan behind this biosensor? Is there really a market? If you're going to develop this, is there really a market that's going to buy this? And so at ZP, we take this holistic view on biosensors, and based on experience, we've actually developed a market analysis group now. So we have a series of scientists and engineers. The client, we talk to the client, we say, what's the vision, what do you want to do? And then we try and establish that they genuinely have a business plan behind this. And if the business plan is not as solid as it could be, then we actually go out there and have to research this business plan, look at the how many diabetics are in the market, or how many patients with renal issues are there, or what's the reimbursement code for troponin I in the United States, and how many troponin I measurements are issued each year, or is it for CRP? So a really important part of biosensor development, science and engineering is great, but it's also the business side of it. Who's going to pay for it? What are the economics involved with it? What's the go-to-market strategy? And so at ZP we have to put together sometimes these business plans for people, and that's why we have this market analysis group. So if I look at the time now, it's 25 past 8 in London time. Thank you, Ali, for your questions this week regarding the microbes. It's appreciated. So yeah, thanks very much. If there's no more questions, then I'll sign off. Please, if you're not a member of the Developer Zone, every Thursday 8 a.m. we do a live streaming for the Developers Zone members, and we'd be delighted to see you there. Okay, thanks very much, and have a good evening.